Thanks a lot for this kind introduction. So I'll talk about, my talk is centered around field calculations as a tool for getting targeting control in brain stimulation, also to estimate the dosage of brain stimulation, simply because we cannot directly measure the electric field, which we induce either by TMS or by transcranial electric stimulation methods in the brain. We have to model it. And when we do that, there are two important questions, namely, how accurate are our models? Can we trust them? And the second thing is, how is actually the coupling between the electric field which we see, which we calculate, and then the neural activity? So what is the mechanism of action in order to be able, this is important, in order to be able to interpret the outcome of the electric field calculations appropriately. And I'll touch on both aspects in my talk. So the first thing is about the sources about one important source of uncertainties in the model, namely the tissue conductivities, and how this influences the results. Then a method for validating our field simulations by using MR as a measurement tool. And finally, I'll give one example on how experiments can be done in order to link estimated fields with physiological effects in order to explain or explore this link between, or this mechanism of action in more detail. So now I'd like to go on. Yes, wonderful. So when you do field calculations using numerical methods in order to be more accurate, you normally start with a head model, and this head model comes either from a structural scan or maybe also some template. The head model is nothing else than a segmentation of the head into the different tissue types, and then from that you create a mesh for your calculation. So you have to discretize your head into the different subvolumes, and the work we're also trying to improve that work is particularly for the skull, and for that I would like to point out to the poster of Ulla Ponti. But in my talk here, for now, I want to more focus on the second part, namely, once you have your head segmented, you have the different tissue types, then you have to basically make an assignment of the ohmic conductivities of the tissues in order to be able to calculate then the field in last instance. And this assignment of conductivities is is difficult because in a way you cannot directly measure them in vivo, so you have to use literature values there, which creates a certain spread, which can be seen here in this graph. Basically, this is a logarithmic scale, and this shows the tissue conductivities. For example, the green one here, this is the lowest conductivity. This would be bone. Then you have some intermediate conductivities. This is gray matter, white matter, and scalp. And then you have here CSF. These are probability density functions which we created based on the data which was out there in literature. And you see that there is quite a big spread. So obviously, compact bone is different from brain tissue, and brain tissue is again different from CSF. But there's quite a bit of overlap simply because the measurements give quite different results, so there's some kind of uncertainty. And the important question is now, how does this uncertainty which we have here actually affect the results of the field estimates which we get? So what we want to do basically is we want to take the uncertainties of the tissue conductivities, which we model as PDFs, as probability density functions, and propagate this uncertainty through our forward model in order to get uncertainty estimates for the electric fields, which we then calculate, basically. And to do that, we use a method called polynomial chaos expansion, which I don't go into further detail here. It's just one possible method which has the advantage of being very efficient, computationally efficient, to solve this. Which means that in our case, we need around 100 forward simulations to generate the probability distribution functions here for our electric fields. What's the outcome then? So we looked at here at 3 different cases. One is TMS of the motor cortex as an example, then 2 TDCS montages, a more focal one, and an unfocal one. And the first thing which you get is, well, you get just average fields out here, which look more or less as you used to see when you have looked into literature on field calculation. So basically when the TMS is relatively focal and stimulates the motor cortex, or in the motor cortex region here, that's the highest field. The focal TDCS field is also very much focused on that one, whereas the unfocal TDCS, yeah, spread along the cortex. So this is basically what we used to see in calculations up to now. The important new thing is now that you also get a variance measure out of that, which is shown here. So basically that tells us how much variance we expect in the electric field due to the uncertainty of our tissue conductivities. And there's already one striking difference here between TMS and the TDCS methods, namely that the variance which we see for TMS is far lower, which means that the tissue conductivities, the uncertainty of the tissue conductivities doesn't really affect our field calculations for TMS a lot, but it has a clear influence on the electric field calculations which we do for TDCS. We can then decompose this variance into different parts which come from the different tissue types, which is shown here. For example, white matter, you see that contributes a little bit to this overall variance. Gray matter contributes a lot, and so on and so on. So when you look into all the different tissue types, then you find that for test methods, for electrical stimulation methods, gray matter, bone, and skin contribute most to these uncertainties. That's in a way just here now another way of showing it. This is now the electric field maximum, and asking the question, how much is this actually affected by the fact that our tissues conductivities which we put into the models are uncertain. So again, you see that the distribution for the TMS case is, so this is normalized, is much more sharp than the distributions for TDCS. So this is in a way bad news for TDCS field calculations. However, we can also ask a slightly different question, namely, how much is the pattern of the electric field actually affected by these uncertainties? With pattern, I mean that we don't care so much about, okay, how strong is the absolute, in absolute terms, how strong is the maximum, but just saying, okay, how reliable is this distribution which we estimate, like that the maximum is a certain position and then it gets weaker here and it gets stronger there again. So how much is this pattern which we calculate affected by our uncertainty? These are again probability distributions, density functions for the relative difference measure, which is a measure for that. Here on the left side you have the reference, and then you see that all the density functions more or less level off here at 0.2. Then the question is, what does our RDM value of 0.2 mean? And for that we sampled just examples from that point here, and you see that basically they are pretty much indistinguishable, meaning that the uncertainties in the tissue conductivities which we have affect the absolute values which we estimate in gray matter quite a bit for TDCS, not for TESS, but the pattern which we estimate, meaning that, yeah, it's stronger here in the center and then falls off and so on. This is pretty robust. So in that sense, we can trust those estimates quite well. Obviously, we would then like to move on and also really improve also the absolute measurements, absolute estimates, and for that we work on measurements based on MR in order to calibrate those simulations which we have. And that's actually in principle quite simple. So when you have a current source and you induce currents in the brain, what the currents will do, they will create a magnetic field in the brain. And this magnetic field will slightly change the magnetic field of the scanner. And by that, it slightly changes also the images which you get out. So the approach there is that you first induce a current with a positive polarity, meaning just it flows in one direction. Then you induce it in negative polarity. You get two different images out. You look at the difference of both, and the difference is proportional basically to the current-induced magnetic field which you have, which you then can use to reconstruct the current flow and the conductivity here. So this is a non-invasive means of really directly validating our electric field calculations. The challenge here is that, well, we can use current limits of maybe 1 or 2 milliampere baseline peak, which also means that the sensitivity which we have in our measurements, or which we need in our measurements, is extremely high. So it has to be better than 1 nanotesla. So we put our subject into a 3 Tesla scanner to measure field changes which are actually lower than 1 nanotesla. We've been working on that in the last few years, going first through systematic optimization of a specific sequence which is suited. It's called SSFP-FID. I don't go further into details. Basically, the systematic optimization was based on block simulations and phantom measurements to determine the best parameters for that sequence. Then it turned out that the best parameters are also those parameters, or parameter settings are those parameter settings which also make the sequence quite susceptible to physiological noise. So we had to fix that in a second step. And the final step is that, well, not only the currents which flow in the brain create a magnetic field, but also the cables here, they also create a magnetic field, which is superimposed then, and we had to correct for that. The results look then like that. So this is a measurement either with the electrodes attached to create an anterior-posterior current flow, or left-right. And this is the magnetic field. So when you have an anterior-posterior current flow, what you expect from the so-called right-hand rule, that the magnetic field is negative on the one side of the current flow and positive on the other side. So that fits already quite well, and it's quite reliable across all subjects. Also left-right is quite reliable, even though there's a little bit more variability. The last thing is then that we said, okay, let's compare that obviously with our simulations. So we reconstructed from the magnetic fields, the current densities in the brain, either measured or simulated, and compared those two. So on average, we have a fit at the moment of 71% R square, which is not too bad as a starting point, I would say. But also interestingly, the simulation systematically underestimated the current strength in the brain by around 24%, which already gives a hint that we can use those types of measurements in the future to really improve our also modeling to get the dosing for tests more accurate. So with that, I want to go to the last topic. Once we have electric field models and we trust them, basically we still have to solve the problem or the challenge that we have to link the colorful pictures here, which are pictures about field strength, for example, to neural activity in order to properly interpret them. So, and this links, I think, still requires a lot of work to establish that link. This is just one example which I give now, a very basic study which we did, where we used TMS on the motor cortex to look at MEPs and motor thresholds, where we just basically rotated the call in 9 different orientations and looked at the motor threshold changes. So this is the average curve here. For the optimal core orientation, you have a lower motor threshold, and then it increases. That's expected. The question now is, how do the electric fields change when we do the same thing? So also rotate the call in our simulations, and is there some correspondence? More specifically, our hypothesis was that, well, we would expect lower motor thresholds for call directions where TMS is just more effective in inducing high field strength in the motor cortex. So basically what we were interested in to see, are there positions in the motor cortex consistently across subjects where we have a good inverse relationship between that shape and those shapes which determine basically how the electric field strength changes with the orientation of the call. And you see it differs quite a bit. So this is, the red one is, for example, at the crown, and the green one is here in the sulcus. So there's quite a bit of change or difference depending on where you are in position. The results were like that quite consistently we saw the highest correspondence, best inverse relationship between field strength and motor threshold here in the upper part of the motor cortex. And when we selected from just the top 10% of so this top strongest fields values here in that region for each of the call position and correlated that with motor threshold changes, then the results were also significant compared to our control condition, which I just now skipped because of time to explain further. Interestingly, so this is where the positions were located. Interestingly, we got very similar results when we took the normal component of the electric field, which means that's the component which either goes 90 degrees in or out of the cortex. Then we got relatively similar results, but this time not on the crown, but on the gyrus lip here, but they're very close to each other. So in general, it seems that the current status is that we can trust the electric field calculations given that we have an anatomically accurate head model for TMS. We can also trust the patterns for TESS. We should still look into further how well the absolute field values are for TESS and whether we have to improve things there. But the MR-based methods already give a quite good fit with their simulations. Also indicate that we can already improve or give a handle on improving our simulations. And finally, this is just one outlook. The final example was just one outlook on how we can establish this link better between electric field simulations and physiological effects, which I think is really important in the future to make most use out of the field simulations. And with that, I'd like to thank you for your attention, and that was it. Thanks.